Coming up next, Sitting at 30 takes you downtown for the weekly luncheon meeting of the City Club of Portland. Live weekly coverage of City Club is produced through the facilities of Portland Cable Access. Now we join the City Club for this week's program.
Thank you. A brief report on our recent membership drive, and today marks the end of that 2-month effort. We have happily welcomed 80 new members, and though today marks the formal close of that membership campaign, I encourage each of us to continue to identify and encourage people in the broader Portland community to become members of the City Club. We look forward to working with all of our new members as they get involved in the City Club, and I'm certain everyone saw the article in the national press over the last few days having to do with specific data that describes the decline nationally in civic engagement, and I think once again City Club of Portland goes against that tide, and we should all be very proud of that. So please continue to get the word out about the City Club, and we welcome new members always. I'd like to acknowledge special members of the club who have recruited new members during the membership campaign. Ann Lyman Foster, her new member is Ellen Proctor. Laura Chardoff, her new member is Heather Kumitz. And Leanne Barnum, and her new member is Chris Tawney. Please thank all of them. Next Friday, please join us for a special program featuring the latest in Oregon energy deregulation plans and learn some more about this much-touted energy crisis. For those of you who still might not be aware, though I think this would be hard to be the case, the City Club website offers each of us access to research reports, past club program presentations, upcoming events, and membership information. Please check us out at www.pdxcityclub.org, and please share that URL broadly. I would also like to bring your attention to a new spring series, which we're calling Uniquely Portland, that features landmark Portland businesses, and this special series will begin April 26th. The series features visits to and presentations from the owners of Rejuvenation House Parts, Urban Wine Works, Powell's Books, and Clear Creek Distillery. All events begin at 5:30 and end promptly at 7. Registration is limited to City Club members and will be limited to 50 participants. Anyone interested should complete the form in this week's bulletin, and please return it to the club office as soon as possible. And now to our program for the day. Our board host this afternoon is Lloyd Anderson, immediate past president of the club and a member of the Board of Governors. It will be his privilege to ask the first question of our speakers. Following Lloyd's question, as always, we will open the program to questions from City Club members in the audience. Please line up behind the microphone even before Lloyd is finished asking his question so that we can entertain as many questions as possible. Thank you. This afternoon, a most distinguished panel will guide us through the once terra incognita of the brain. I occasionally have synaptic lapses, so if I pause for a painfully long time, please, any of you, feel free to commandeer the microphone. I must also admit that until this afternoon, the closest I had personally come to brain research was my preparing for the role of the Scarecrow in a summer camp production of The Wizard of Oz, but I will blessedly spare you those stories. Over the past few years, many findings have literally stood neuroscience on its head. The now recognized plasticity of the brain, for example, and findings that brain cells regenerate in many primates closer to us on the evolutionary scale than mice, and that nerve cells regenerate in a portion of the brain related to memory and learning. These are fantastic breakthroughs, but they also raise fantastic and profound questions well beyond the world of therapeutic medicine. Questions that challenge each of us to think about the philosophical, moral, and ethical underpinnings of what it means to be human. Questions about character and identity. Currently, over 1/3 of the activity at Oregon Health Sciences University is involved in some aspect of neuroscience or neurological research, education, and care. In addition, the new Neurological Sciences Institute, built on OHSU's West Campus, represents a significant commitment to accelerating research efforts in these areas, both regionally and nationally. It's now my privilege to turn the program over to Dr. Peter Kohler, President of Oregon Health Sciences University, and his esteemed colleagues, Drs. Ed Neuwelt, Dennis Bourdette, and Paul Cordo. Dr. Kohler.
Thank you, Harriet. I spoke to the City Club 6 years ago in February of 1995, and as you can imagine, a great deal has changed since that time. Then OHSU was in the middle of a public corporation debate, a policy that was overwhelmingly adopted by our legislature. That move, we think, was very successful, literally saving OHSU. Since that time, we have increased our number of employees by over 50%, about 3,500 additional people to over 10,000 employees, including faculty at OHSU. And we have been developing, importantly for today's topic, into a national research leader. We are today a much larger, yet leaner, more entrepreneurial institution. And our growth has been driven in large part by our research. The quality of scientists at OHSU bring NIH money into Oregon, $168 million of awards for the year 2000, money which enhances our research programs. In fact, we're in the middle of a campaign to utilize the great value created by our research programs to develop the biotechnology industry here in the state. We call that the Oregon Opportunity. We believe that Oregon should have a much greater bioscience, biotechnology presence. We have several strong research programs. You probably have heard a lot recently about our oncology programs and the sensational work being done by our scientists such as Drs. Drucker, Nichols, Blanky, Bagby, and others. We believe that our neuroscience team, with a number of exceptional research and clinical activities, as you can see from a chart, which I think is over in that corner there, is poised to achieve the same kind of recognition for its work. And that work is the basis for the presentation today. The subject at hand is the brain. Research advances have been occurring rapidly in the neurological sciences. The brain is by far the most complex of all human organs. It contains nearly 1 trillion nerve cells, each one interacting with the other in intricate and mysterious ways sometimes that make it possible for us to see, to hear, to breathe, touch, smell, sleep, walk, you name it, feel. The brain is perhaps the most important and least understood of human organs. Indeed, the human brain remains the last great frontier of science. We are only beginning to unlock its mysteries. For example, recent research suggests the possibility to add new brain cells in adults, a concept believed impossible just a few years ago. Neurological disorders include diseases of the brain, spinal cord, and peripheral nerves. Ronald Reagan, Annette Funicello, Michael J. Fox, Christopher Reeve, these are just a few of the famous people who suffer from disorders of the brain and nervous system. But of course, none of us is immune. In fact, as we conquer cancer and heart disease, we are seeing a rise in the number of people with Alzheimer's disease, Parkinson's, MS, stroke, and other debilitating diseases of the nervous system. Each of us, as individuals, face a 75% probability of suffering a neurological disorder. 75%. And in total, more than 50 million Americans now suffer from over 1,000 neurological diseases. Collectively, these maladies cause more hospital stays and disabilities than any other group of illnesses, including heart disease and cancer. In the U.S. alone, the annual cost of medical treatment, long-term care, and lost productivity tied to disorders of the brain and central nervous system is a staggering $600 billion. These losses are magnified by the intangible costs incurred by the families and society. Some neurological disorders, such as mental illness, are often seen as environmental, but in fact have an organic component in most instances. Researchers now know, for example, that depression is related to lowered levels of serotonin. Similarly, substance abuse is not frequently considered by the general public to be a neurological problem, when in fact addiction is not only a specific form of mental illness, it is the most common mental illness in the United States, afflicting as many as 1 in 10 Americans. For young adults, substance abuse is the most common medical ailment. We in Oregon have particular problems with methamphetamines and heroin addiction due to the unfortunate ease of acquiring these deadly drugs. Today we have cures for cancer, some developed at OHSU just recently, but as of today there are no known cures for any of the common disabling diseases of the brain and nervous system. But through research there is great hope. Today we understand the neurochemical basis of addiction much better than we ever have. What's more, researchers have identified the genes underlying neurological disorders such as Alzheimer's, Parkinson's, Huntington's disease, and ALS. These discoveries have shed new light on the development of these disorders and have suggested treatment options for further investigation. In fact, some of the scientific community believe, based on research progress to date, that there will be a cure for some of these neurological disorders in the next 3 to 5 years. We believe these cures could easily come here in Oregon. We have great strength throughout the state in the neurological sciences, including our sister institutions throughout Oregon. We have a number of prominent neuroscience programs at OHSU. These range from basic brain research, in which the fundamental principles underlying brain organization and function are elucidated, to clinical brain research, which applies these principles to an understanding of how neurological diseases affect the functions of the brain and the nervous system. While we are deservedly proud, I think, of our research and clinical programs in cancer and other diseases, we should note that nearly 1/2 of the NIH support coming to OHSU is in the neurosciences. A number of important research advances have been made at OHSU, including the following discoveries. Brain tissue does not, as previously believed, automatically shrink with age. There's been a finding of an establishment of a connection between a common immune gene and the early onset of Alzheimer's. An invention of groundbreaking techniques that allow researchers to videotape cell communication. Very impressive. Identification of key brain cell communication links that may help researchers understand such conditions as stroke, epilepsy, and head trauma. The development of deep brain stimulation as a treatment for Parkinson's disease. We've had the establishment of one of 2 NIH-funded Alzheimer's research centers in the Northwest, and we have one of 6 VA research centers for Parkinson's throughout the country. We have had development of technology to allow the efficient delivery of drugs and genes to treat brain tumors and genetic diseases of the brain. At OHSU, we've also achieved a number of firsts. The first to discover 4 of the 5 known dopamine receptors, proteins involved in Parkinson's disease and some forms of mental illness. We are the first to test a new plant derivative from cat's claw as a treatment for Alzheimer's, and the first to test another plant derivative, ginkgo biloba, for its ability to prevent Alzheimer's. The first to develop and test a vaccine for MS, and you may hear more from Dr. Bourdette on that topic. The first and only allopathic medical center to establish an NIH-funded research center with partners dedicated to studying non-traditional or alternative therapies for the neurological disorders. Again, these are just a few of the many discoveries made at OHSU or activities ongoing there, and you can see why we're optimistic for future breakthroughs here in Oregon. At OHSU, research and clinical care are conducted in a number of disease-specific centers, such as the Parkinson's Center of Oregon, the Oregon Stroke Center, the Oregon Aging and Alzheimer's Disease Center, and the MS Center of Oregon. We have 3 top-flight research institutes engaged in broad-based research: the Center for Research on Occupational and Environmental Toxicology, or CROIT, the Vallum Institute, named for the generous endowment by Tektronix co-founder Howard Vallum, and the Neuroscience Institute, or NSI, which came to us from the Legacy Health System. Each is engaged in multiple basic science and clinical research projects related to the brain and nervous system. These are just a few of the assets OHSU employs in the fight against neurological disorders. Again, I would refer you to the chart which shows a number of component parts to our activity. They are arrayed across our multiple missions of teaching, research, and clinical care. At OHSU, we talk about critical mass, and we're close to reaching that in the field of the neurological sciences. 3 men that are with me today are reflective of breadth and depth of excellence in neuroscience research at OHSU. Dr. Ed Neuwelt will discuss gene transfer for neurological disorders. Dr. Dennis Bourdette will describe the immune attack on the central nervous system. And Dr. Paul Cordo will discuss efforts to rehabilitate people with neurological problems. Many of you may have heard about OHSU's initiative to achieve a major leap forward in our research programs. When you hear from these 3 today about the exciting research they are conducting, I think you'll understand why we feel the time for progress is now. Dr. Ed Neuwelt is professor of neurology and associate professor of neurological surgery at OHSU. He received his medical degree from the University of Colorado School of Medicine. Dr. Neuwelt served a surgical internship and neurosurgical residency at the University of Texas Southwestern Medical School in Dallas. He was also a neurological fellow at Queen Square Hospital in London. His special area of interest is on the therapy of brain tumors and central nervous system diseases. He is a rare combination, I can tell you maybe the only, of a practicing neurosurgeon and a basic molecular investigator. Dr. Dennis Bourdette, who you'll hear second, is interim chairman and Swank professor of neurology at OHSU, as well as a staff neurologist at the VA Medical Center. He is also co-director of the Multiple Sclerosis Center of Oregon at OHSU. He is a graduate of the University of California, Davis School of Medicine. He completed his neurology residency at OHSU and a fellowship in neuroimmunology at the Portland VA. His area of interest is in the immunopathogenesis treatment of MS and other neuroimmunologic disorders. His department, by the way, ranks number 11 among neurology departments in the country in NIH awards as of the year 2000. Dr. Paul Cordo is director of the Neurological Sciences Institute and professor of physiology and pharmacology at OHSU. He received his undergraduate degree in biomedical engineering at Rensselaer Polytechnic Institute. He received his PhD in physiology at the State University of New York Upstate Medical Center in Syracuse. His area of research interest is motor coordination and proprioception. It's often been said that we are entering a golden age of medical research. Between the completion of the Human Genome Project and the dramatic and ongoing increases to the NIH budget, funding-wise, the promise of progress here has never been brighter. We at OHSU are trying to capture the breadth and economic benefit of the research boom for all Oregonians through the Oregon Opportunity. As I said before, we're nearing a critical mass in terms of our scientists, but we're literally running out of space in which to house these individuals. Oregon can and should be one of the national leaders in neuroscience research, but we need to work with the state, the city, federal government, and public-spirited citizens involved in the clinical research because, in fact, to test these cures, we need people who are willing to participate in the testing. If we work together, I believe we can find the next generation of cures for these debilitating and deadly diseases of the brain. And we can do it right here in Oregon. Now I would like to ask Dr. Neuwelt to come to the podium and present what I think you'll find to be very interesting findings. He'll be followed by Dr. Bourdette and Dr. Cordo in that order.
I wish to express my appreciation to the City Club for letting Dr. Kohler and the rest of us come down, and to Ms. Watson, because I think what Dr. Kohler just said is really the reason we're on the Hill, is to try to combine patient care, research, and teaching both on and off the Hill, and that's really why I'm here. So it was exciting to be asked by Dr. Kohler to do this. The topic which I'd like to talk about in just a couple minutes is gene therapy of the brain. Now, Dr. Kohler pointed out that we've just finished the decade of the brain nationally, and as trying to treat the brain. And so clearly the brain in a lot of ways is the last frontier, as he pointed out. But there's an Achilles heel to that. And that's the sort of small area that I try to focus my attention on, and it's the blood-brain barrier. The brain is an electrical organ. All its communications are with electrical signals that then go to certain chemical responses, and it functions like a mainframe computer. Thereby, it has to have its own air conditioning system. And if you mess around with it, it doesn't work so well. So if you have a meal like you do now, you'll have certain substances in your blood. If you go out tonight with your kid to McDonald's, you'll have other substances. And what happens is, if those substances were to get into the brain, the very delicate molecular environment of the brain cells would be disturbed. And therefore, Darwin showed that nature has developed what's called the blood-brain barrier. And the blood-brain barrier keeps virtually everything that you eat out of the brain, with the exception of a very few nutrients. There's a handful of carriers that take specific amino acids and nucleotides across. Sugar, it goes across very well. The brain really likes fatty acids and eats that up very well. But virtually everything else is kept out. And it's done by the fact that the cells that line the blood vessels in the brain, they're called endothelial cells, are glued together by things called tight junctions. And those tight junctions are great to allow us to have the variety in eating that we do, because they keep virtually everything else out, and the environment of those electrical brain cells incredibly constant. But that's a problem. And it's a problem if you have diseases of the brain. And the fact is that most drugs, proteins, and genetic vectors, which is what I'm going to try to talk to you about very briefly, are kept out. Because this blood-brain barrier is so effective that if you've ever seen spinal fluid from someone having a spinal tap, it's as clear as water. And if you have a test tube of spinal fluid and a test tube of water next to each other, you couldn't tell the difference. On the other hand, obviously, if you took a syringe full of blood and even took out the red and the white cells, it's a thick, proteinaceous, straw-colored fluid. So the blood-brain barrier is incredibly effective in maintaining the environment necessary for this mainframe computer to work. But it's a problem because it makes it hard for us to treat diseases in the brain. And although industry around the United States during this decade of the brain has spent billions and billions and billions of dollars on making all kinds of fancy new molecules to treat brain disease, they've spent almost nothing on how to deliver them. So there are a few of us in the world that are interested in the delivery business of getting drugs, proteins, and viruses to the brain. And it's very important to do this in a fashion where we can go from the test tube to animal models to patients and back. And that's what's so nice about the Hill, is the fact that we really, in proximity, our laboratories and patients are in the same place. Now, in the world of gene therapy, there are really two kinds of gene therapy. There's germline gene therapy, where you try to change inheritance. And I'm not involved in that at all. So I think that's an important distinction. I'm involved in what's called somatic cell gene therapy. That's basically making genes to treat a given individual with a given disorder, which will not be passed on. And in the brain, it's very difficult to get genes into the brain because the average drug, if you remember from high school chemistry, has a molecular weight of about 400. Particles of genes are in the millions with regard to the molecular weight. So they're very large molecules. And in fact, they're so big that we like to package them in crippled viruses that normally cause infection, but we take out the replicative part of the virus. So it's very important to separate, in treating neurologic disease, somatic cell versus germline therapy, and the fact that we use vectors. Now, in our research, we use two kinds of models. We have one model, which is what I do clinically mostly, is take care of brain tumors, where we're trying to deliver genes that will selectively kill brain tumor cells and not normal brain cells. And that's called more focal gene therapy. And the second kind of gene therapy that we're involved in is treating global neurodegenerative disease. And I would like to close my comments with just a comment about that model. The model that we use is a naturally occurring disease in a cat model that was brought to me by a cat breeder who has Tay-Sachs disease. Now, Tay-Sachs disease is a very rare disease. And in fact, it's the only human disease where genetic testing has dramatically decreased the incidence. But that disease causes the buildup of a lipid. And that lipid leads to what's called programmed cell death. So it's a wonderful model of a neurodegenerative disease in which there's programmed cell death, just like in the other diseases that Dr. Kohler talked about, Alzheimer's and Parkinson's. And it's interesting because there's a single nucleotide out of 1,700 that was left out, which causes the disease. And so what we've done in our laboratory is to clone that gene from this animal model and are in the process of delivering it across the blood-brain barrier by taking concentrated sugar solutions that we inject through the arteries of the brain, the carotid or the vertebral, that make those cells that line the brain, the endothelial cells, shrink just by osmosis. And when they do, the tight junctions open, and we can put those viral vectors across the blood-brain barrier. And in fact, we're currently, with NIH funding, taking iron particles the size of these viral vectors and looking at them in patients in early trials going toward global neurodegenerative disease. Thank you.
So I'm Dennis Bourdette. I'm a neurologist. Dr. Neuwelt's a neurosurgeon. Biggest difference being at Thanksgiving, he's allowed to carve the turkey. And I am a turkey. It's a real pleasure to be here. I do have to admit, though, that I formerly thought that the City Club was something the mayor used on her political opponents. But I hear that's not true. I wanted to reiterate some comments that Dr. Kohler made. Over the coming decades, we are going to see an epidemic in chronic neurologic diseases. And the reason for that is because we're living longer. And all of these diseases, particularly Alzheimer's, Parkinson's, and stroke, the risk of developing them rises with age. So while our politicians are worrying about the aging of America and its effects on Social Security, and our financial planners are helping us to make decisions about our own savings for retirement, they're emphasizing that we're going to be living longer. What we are not doing really as a nation, I believe, is emphasizing that part of this is not just worrying about money, but part of this should be worrying about a looming epidemic of neurologic diseases. If you live long enough, you will develop a neurologic disorder. It may be Alzheimer's disease, it may be Parkinson's disease, it may be a stroke, but you will develop it. So this is something of vital importance to the future of our nation. Now, fortunately, like the cavalry coming to the rescue, we do have the tools in hand to conquer these diseases of the brain. Just a decade ago, neurologists such as myself believed that we were decades away, literally decades away, from developing effective treatments for the common chronic neurologic diseases. But today, we already have new treatments for many of these diseases, and we are realistically looking forward to developing newer, more effective treatments and perhaps even cures over the coming decade. And I want to tell you that OHSU is poised to be one of the nation's leaders in neurosciences research. Now, what are the tools? The tools that we have at hand to conquer these diseases come in advances in our understanding of the genetic basis of these disorders, advances in understanding how the brain works and how it responds to disease, how it works in the living state, and very importantly, with our ability through magnetic resonance imaging and other imaging techniques to image the brain in health and disease and to image the response of the brain to new treatments. So while we face a growing epidemic of chronic disabling brain diseases, we have in hand the tools to combat these diseases and to develop new therapies. It really is just a question of collective will. Are we willing as a society to make the necessary investment in research that is needed to develop new treatments for these serious illnesses? Now, I want to tell you about our work with one neurologic disease, multiple sclerosis. Now, MS affects nearly 500,000 Americans and over 5,000 Oregonians. It strikes mainly young and middle-aged adults, peak times for work and for raising a family, and too often it disables patients, rendering them unable to be gainfully employed, unable to participate in the joys of raising a family. MS is a disease in which specialized white blood cells attack the nerve fibers in the brain, spinal cord, and nerves to the eyes. And as a result, it causes a variety of problems, including weakness and paralysis of the legs and arms, loss of control of the bladder, cognitive and emotional difficulties, and even blindness. Now, prior to 1994, there were no treatments, absolutely none, that could slow the progression of MS. However, since 1994, through medical research, including research done here in Portland, we now have 4 FDA-approved treatments for MS that partially control the disease. This is something we really did not dream of occurring in 1990. Now, about 15 years ago, we established at OHSU the MS Center of Oregon. This is a center that brings together physicians, clinical and laboratory scientists, nurses, and other personnel devoted to providing state-of-the-art care for patients with MS while conducting research into the cause and cure of the disease. We currently have a robust research program with over $2 million a year in federal support coming to Oregon to support MS research. I want to tell you briefly about 2 new treatment approaches that were developed exclusively at OHSU. I need to tell you that I said white blood cells cause MS. Every single person in this room, whether you have MS or not, have these white blood cells that can cause MS floating in your bloodstreams. We all have them. The difference, though, between those of us that don't have MS and those that do is that we have other white blood cells that can effectively control and keep those cells under check. And one of our major missions have been to develop ways to control the white blood cells and in particular to augment the body's natural ability to control them. So the first approach that was developed at OHSU is called TCR peptide vaccination. Quite simply, this entails vaccinating patients with a piece of protein from the surface of the disease-causing white blood cells. The vaccine then stimulates protective white blood cells that can control the cells that cause MS. It's not a vaccine that's designed to prevent MS, but rather to treat it once it's developed. So it's analogous to attempts to develop vaccines for certain forms of cancer. This treatment was first developed in an animal model of MS by scientists at OHSU. The initial trials in patients were then conducted in Oregon through the MS Center. And these early trials indicated that if we could get a strong, robust immune response, patients would be improved. But unfortunately, the current vaccine does not vaccinate all patients, and we are working with a biotechnology company based in San Diego to improve the vaccine and are conducting another clinical trial at 6 sites in Oregon and Washington. The fact that we're partnering with a company in San Diego may be an issue we'd like to return to at the questions and answers. A second treatment is the development of another type of protein that also holds the potential for controlling the disease. There's a certain gene called DR2, which is associated with MS in about 60% of patients. This gene makes a protein that is important to turning on disease-causing white blood cells. Scientists at the MS Center have genetically engineered a new form of this DR2 protein that can turn off the disease-causing white blood cells. This treatment works in an animal model of MS and in the test tube. The next step is to begin testing it in humans. Because this involves genetically engineered proteins, it will be quite costly to begin the human trials, and we are therefore in the process of looking for biotechnology companies to partner with to finance the development of this treatment. I think it's worth mentioning again that so far all the potential partners are based outside of Oregon. Both of these treatments are novel and were developed exclusively at OHSU. They were made possible because we assembled a team of laboratory and clinical scientists who worked together closely with one aim, namely to develop new treatments and ultimately a cure for MS. I think similar approaches and similar teams are at work at OHSU trying to conquer Alzheimer's disease, stroke, Parkinson's disease, and other neurologic disorders. OHSU is a unique resource, and particularly for patients suffering from chronic neurologic diseases, our research and clinical programs offer hope for a better future.
Hi, I'm very happy to have the opportunity to talk to you about my research. I'm a PhD. My name is Paul Cordo. I'm the director of NSI. As a PhD and not a clinician, I guess that allows me to pluck the turkey. The research that I do and that most of the neuroscientists at NSI do is clinically related, and I also do some bona fide clinical research. Begins from an interest in how things work normally, and so my work in studying a basic area of neurophysiology has led into, not by design, into an area of clinical research that impacts individuals that have had strokes. In the U.S., there are about 500,000 people that have strokes every year. And most stroke victims who survive go through a process of recovery that lasts typically from about 6 to 12 months. And this recovery can be substantial. In fact, in some people it can be complete, but there exist between 4 and 5 million American citizens who are permanently, at this point, disabled, severely disabled by stroke. Now, the most prominent disability associated with stroke is the loss of movement, and happens on the opposite side to the side of the brain that has the injury. And in my lab, we've been working on a new method for helping chronically disabled stroke patients recover the ability to move. Now, my lab investigates the sense of proprioception, and this is, or kinesthesia, and this is the sense of your body's position in space and the sense of movement. And it's a sense that is processed almost completely on the subconscious level. It's probably why it's not included in high school textbooks. But it is, in fact, a major sensation and essential for coordinated movement. And the sensation of the sense of proprioception comes from a variety of different receptors, sensory receptors, that are located in the skin, in the joint capsules, in the tendons, and in muscles. And perhaps the most important of these is called the muscle spindle, which is a receptor. It's relatively large, lives in muscles, several hundred typically in a given muscle, and it measures the length of the muscle. It fires in response to the length of the muscle to stretch, and that information gets to the brain, and the brain translates that into an understanding of the position of the joint. Now, one thing that's peculiar about muscle spindles is that they're particularly sensitive to very small movements, precise movements. So in the laboratory, we can take advantage of that, and we can stimulate muscle spindles with vibration. We apply vibration with a vibrator to the tendon of the muscle, and each tap produced on the tendon by the vibrator can elicit a discrete electrical pulse or action potential from the receptor. And so by changing the frequency of the vibration, we can send many different kinds of signals to the brain related to movement. So if I were to take a vibrator and apply it to one of your tendons, you would experience a sensation of motion at the joint crossed by that tendon, even if there was no motion actually taking place. So in my lab, we've done basic research on proprioception and movement coordination for many years using this vibration technique, and then recently we've begun to apply it to the treatment of stroke patients. Now, let me digress just a moment here. There are two parts of the brain that are particularly important for producing movement. One is the sensory cortex, and this is the area whose nerve cells receives the proprioceptive information and touch information from the body. And the second is called the motor cortex, and that's the output, the part where the output neurons go to the muscles and actually produce the movement. Now, both of these parts of the brain are organized in terms of maps of the body. So, for instance, the part of the sensory cortex that receives touch and proprioception information from the index finger up here is situated adjacent to the one that receives it from the middle finger and from the thumb. Now, when most people recover from stroke, circuits in undamaged parts of the brain presumably take over some of the functions for the damaged tissue. And the sensory cortex and the motor cortex exhibit this ability to reorganize rapidly, on one hand to changes in sensory information, or on the other hand to changes or repeated types of movements. So if I were to take your finger, and one of your fingers, say your index finger, and I was to anesthetize it, and I could record from the brain cells in your sensory cortex, the cells that normally respond to sensory information from the index finger, within a half hour or so, many of them would begin to respond not to the index finger, but to the thumb and to the middle finger. And after the anesthetic wore off, after a while, this reorganization would reverse itself. And similarly, in the motor cortex, if you were to perform a task, repetitive task, let's say you were to hold a ball bearing between your middle finger and your thumb, you were to roll that around in some way, after about an hour or two, what you'd find is the output neurons that normally control the muscles of the index finger, which is not involved in this task, would begin to activate muscle controlling the middle finger and the thumb. So the brain tissue here is very plastic, especially that part which is involved in the control of movement. Now, because of the way the circulation is organized in the human brain, many strokes do not affect the sensory cortex or the motor cortex, yet these people have extremely impaired movements, or they're paralyzed. The part of the brain in many of these cases which is damaged includes the main connection between the sensory cortex and the motor cortex. And so we think that the reason that people lose the ability to move, even though the sensory and the motor cortex isn't affected, is that in order to move a part of your body, you have to sense it. And without that connection between the two, you're not able to sense it. And we also think that the extent to which individuals recover the ability to move after a stroke depends on how well these two brain areas are reconnected during the plastic changes that take place during the recovery process in the year or so that follows the stroke. So in my lab, we've been studying how people who, long after the recovery from a stroke is complete, can be retrained to recover the ability to move. And with increased mobility, these individuals then might be able to walk, they might be able to feed themselves and to carry out the normal activities of daily living. So the way we do this is with a computer-controlled machine that we've designed and built in my laboratory. And so far we've built about 10 of these, a number of them for the ankle, for the elbow, and for the wrist and fingers. We put the machine into the patient's home, and they use it for 30 minutes a day for 6 months. And the individual sits in a chair and places the appropriate joint into the machine. And then a motor on the machine moves the joint back and forth. And the individual's job is to assist the motion of the joint imposed by the machine. And they can start this whole process very easily with a computer touchscreen. They just push a button with their good arm. So it's designed to be a fairly user-friendly device, and many stroke patients can use the machine without any help from a caregiver. Now, trying to assist the device is very difficult for many stroke patients. They don't have very much movement, and so we provide biofeedback on the computer screen to tell them how well they're assisting. In addition to this, at the same time, there are two vibrators, one on each side of the joint in this machine, and as a particular one, the muscle is lengthened on one side or the other, a vibrator vibrates the tendon across there. So the idea behind this procedure is to enhance the brain activity, first of all, in the sensory input area of the brain using the vibration. So we vibrate at a high frequency, and this enhances the sensory activity in the sensory cortex. And at the same time, the stroke patient is trying to assist the machine, and by doing that, they're activating the nerve cells in the motor cortex that are projecting down to the muscles. So by activating these two functionally related areas of the human brain at the same time and over, we hope to develop the new connections between these two areas and allow people to recover movement. Now, the studies that we've done so far are still somewhat preliminary, but our results are pretty promising. If the sensory and the motor areas of the brain are more or less intact, we can usually produce significant recovery of movement, both in the leg and the arm. And in addition, the muscle mass increases significantly, and also spasticity or rigidity of the joint, which is another characteristic of stroke, is diminished. And what we found is that these beneficial effects appear to be sustained after the period of the 6-month treatment is over with. So we're very hopeful for the future in this research. So I'll just end by pointing out that if I hadn't been working on basic research using tendon vibration and these other techniques, I probably would have never thought to use them to apply them to stroke patients. And I think that's a common experience in biomedical research. So first comes a breakthrough in understanding of some basic problem in how the brain works normally, and then comes an idea about how to apply that. Thank you.
Peter, it's always impressive to have your organization up here, and certainly very humbling. I'm a volunteer for Alzheimer's up at the university. My big fear in that, of course, is that they'll examine my brain and find nothing. But I do have a question with reference to the university and its financing. And it goes like this. You mentioned federal funding for a substantial amount of it. Presumably there's also a substantial amount of state funding. First, kind of what share of your total income is from those two sources? And secondly, what are your expectations and hopes out of those institutions for the future? Do you get a good reception? Is it hostile? What kind of success have you had, and what are your hopes?
Well, we have a public mission in education, which we view as a partnership with the state. The state currently funds us at the rate of a little over 6% of our total budget per year. We are a very large institution, as I indicated earlier, and we receive a lot of our financial support from a variety of sources, including our research programs, which are now 3 times the size of the state appropriation, if you want to put that in context. A lot of our revenue also comes from the clinical programs that are ongoing at our institution as well. I think we receive a good reception from the state. We are looking at this Oregon opportunity as a question of investing some of the tobacco money into research programs that not only could enhance our research at OHSU, but also serve as the basis for biotechnology in the state. Dr. Bourdette alluded a couple times to biotechnology companies in San Diego. Certain parts of this country have done an exceptionally good job in establishing the right culture for bioscience or biotechnology It is here in Oregon, but in a very, very limited amount. And I think as the excitement of the advances in biomedical research in the neurological sciences and others goes forward, it's important for us to try to establish that here, not only because of the humanitarian and health benefit that can occur, but also, frankly, it's a very great stimulus. It'll be a big part of the economy somewhere, and we believe that somewhere should include Oregon. So the NIH budget, as a large part of our research funding, is in the process of a doubling. At least there is a campaign to double that over a 5-year period, and we're in the middle of that 5-year period right now. So this is coming right as the Genome Project is coming to the conclusion, and to me this is the best time ever in history for biomedical research, and the federal government is looking at bringing that federal budget for NIH along, the main funder for biomedical research, at this time, and we believe that we should be able to participate in that. So the Oregon Opportunity is an effort to take some tobacco money, build out capacity, and utilize that to continue to attract NIH money to OHSU.
I'm hoping that you can tell us what the progress is on research in the heredity of MS and any tests that might be available for susceptibility to MS.
Sure. MS, like a lot of chronic diseases, not just anybody gets it. You have to be genetically predisposed to development. But it's not a strict genetic disease in the sense that if you have it, half your offsprings are going to have the disease. There are multiple genes that predispose people to developing it, and there are probably also genes that are protective for MS. There's a large international study trying to define the genes that increase and decrease the risk of MS. It's being driven by investigators at UCSF, although we're participating with that, helping to identify families. Thus far, the only gene that's been clearly associated with an increased risk of MS is this DR2 gene that I mentioned earlier that 60% of patients with MS have, only 20% of the general population have it. But I believe over the next 5 years we're going to see several other genes being identified, and the big hope there, of course, is that it's going to lead to new treatments, because if we can identify genes that are making proteins that either promote the disease or alternatively proteins that help to inhibit the disease through genetic engineering, one could actually make these proteins and use them as supplements as a treatment. So that's where we're right now. But I think over the next 5 years we're going to see a lot of new information coming out about the genes that predispose and perhaps protect against MS.
Ray Polanyi, a City Club member. I have an ethical and moral question that I hope will be useful. Let me paraphrase Albert Einstein, who said that unless we advance dramatically in social relations, he foresaw great peril for the future of humanity. Technological breakthroughs accelerate, yet the misuse and misapplication of technology for short-term gain is very evident. The question is, will brain research help us catch up socially? And will we?
Dr. Kohler, would you like to take that?
I thought we were going off into the ethics of gene transfer, but which is another very big area, as you know. I think so. I think the more... I have just finished working on a report on the quality of long-term care in this country. And the incidence of dementia that occurs in people over age 65 is truly frightening. And we think that using the brain protects it. There are a lot of issues around that need further study. But I think the greater degree to which we can normalize function in individuals with neurological conditions through the techniques that you're hearing about today, I think the better off we'll be in society. Because if we're looking for ways to live out a normal life, and we think there is probably a defined span during which you live, to enjoy the later years, the brain function should be as normal as possible. And I think that aids the social adjustment. We would very much like to find ways to prevent Alzheimer's or reverse it if it occurs. I think all of these will be to the benefit of society at large.
Can I add something to that? Dr. Kohler mentioned that drug addiction is really a neurologic disease. There's genetic influences on that, and there are chemical changes related to that. And I think if we were able to find ways to prevent drug and alcohol addiction that have tremendous social consequences because of the enormous havoc that those conditions bring to society and to families and individuals.
I'm Nancy Lipset from the City Club, and I wanted to know, Dr. Bourdette, has your research added to your knowledge of Parkinson's disease, and if so, what can you tell us about it?
Well, my answer to that question a few years ago would have been no. Mm-hmm. But what we've seen developing over the past few years is a recognition that the brain only has a limited number of ways to respond to injury and to diseases, so that a lot of what we learn about MS may well be applicable to other neurologic diseases and vice versa. I think that one of the major issues in Parkinson's is what's causing the nerve cell damage in Parkinson's. We now recognize that nerve cells and the nerve fibers themselves are dying in MS. And it may well be that in both cases there's changes at the genetic level and changes caused by small molecules called free radicals that are damaging the nerve fibers and cells. So that I think advances in one disease may ultimately help another. Another area that we're particularly interested in trying to open up and develop at OHSU is the development of a neural regeneration program. There is a variety of approaches that are being applied in the laboratory to MS and Parkinson's disease, finding ways to promote both protection of nerve cell death and actually regeneration. And I think that's going to be a major growth area of research, and one that I think OHSU should be involved with.
Thank you.
Deborah House, City Club member. I first want to thank Dr. Bourdette and some of the other folks in your department. I was one of the first people in Oregon to get beta-seron, and if you ever need a poster child, it's a miracle drug. You started earlier, Dr. Kohler, by talking about the fact that you're running out of space and that you're going to need help from the city, amongst others. What kind of help do you need?
That's a great opening. We have a 30-year strategic planning operation, which is way out in the future compared to the way most places have to plan. And frankly, we anticipate, we have added, for those of you who may not be aware, 1 million square feet of space over the last decade at OHSU. That's allowed us to grow our research programs and our other programs quite handily. On the other hand, if you have been up to what we call the Hill recently, you realize there are some problems of access there, finding your way to start with and then finding a place to park later. We need to be able to expand, and we expect over the next 30 years to build out a net 2 million additional square feet. So we're looking for space where that can occur. We have a number of opportunities available to us. We have space actually on the Hill that could be utilized if the access were better. We have the West Campus, which is where the Oregon Regional Primate Center is located and where the Neuroscience Institute and the Vaccine and Gene Therapy Institute are now located. And you probably are aware that we will be merging with Oregon Graduate Institute this summer and forming a partnership with them to take advantage of their tremendous expertise in computer science and engineering to move forward in biomedical research as well as the high-tech activity they already have. In addition, there has been some discussion with the city about the land on the riverfront in North Macadam, and we're looking right now at feasibility for all these options about what we really should be doing. But we do know that to continue to make the progress that we have before, we need to continue to add space and the premier scientists who bring their money with them. I think that's an important concept. The NIH budget is based on merit, And if you can recruit the very best minds to come to OHSU, they will bring their research money with them, which helps the state and which helps our research programs. So we will be needing to expand, and exactly where all the expansion will occur is still not determined yet. We're looking at the feasibility.
Unfortunately, that will have to be the last word for the formal part of the program, and I thank all of our participants today. I would also just like to suggest that in setting some of the excitement that's happening on the Hill into a broader public policy framework, all I kept thinking of was our ability to be able to push down some of this excitement into K through 16 so that we get a partnership with some of the young people who are coming through the system, both in our K through 12 and then in our undergraduate work, that this science has meaning well beyond a strictly structured curriculum. Thank you very much. We're adjourned.
Even though we're adjourned and off the air, let me make a final comment that relates to that. For us to be able to make the progress that we think is going to be appropriate on the research front, it's critically important that K through 12 and K through 16 be very involved, particularly in math and science. It's very important that that be done appropriately and adequately in the curriculum. So that is to have the prepared workforce available in Oregon where we want this to be, that's a key part. I just wanted to add that on to the end even though we're off the air.
There's also a brief new member orientation in the lounge. Thank you.
Thanks, appreciate it.